Hello dear friends, I hope you all are doing fine. In this video, you will get the solution of the question paper of the CUET PG exam held in the year 2022 for the subject Life Sciences. This video is very important for you if you are preparing for the entrance examination. So watch this video till end. Myself Dr. Tripti Malika Ahuja. I am your tutor, guide and mentor for the subject Biological Sciences, Life Sciences and Microbiology. In the I button here and in the description box below, you will find the link for the playlist which has number of videos which will help you in the preparation of your entrance examination. If you are new to the channel, then do subscribe it and also press the bell icon so that you can get the notifications of all the new videos. So I will read you the question number 26. Actually, this is the part B of the question paper. The part A has 25 questions which are based on the general knowledge, English, reasoning and aptitude. The part B is the subject based part in which there are 75 questions. So I will start reading the questions. And as I start reading the questions, I suggest you to take out the paper and a pen as I will read the question and then I will tell you the options also. Out of these 4 options, only one option is correct. The option which you think is correct, you can write on the paper and then I will give you the correct answer with an explanation also. If you're having any doubt or you disagree, you want further explanation, then you can write in the comment section. I will reply to each and every comment. So question number 26. Which of the following technique is used for tracing change in intracellular location of proteins and metabolites? Option A, chromatography. B, DNA footprinting. C, DNA and labeling. D, pulse chase experiment. I hope you have got the correct answer, and the correct answer here is the option D, which is the pulse chase experiment. It is used in biochemistry and molecular biology. The cells, they are exposed to a labeled compound which is called pulse for a short duration, and then it is exposed to the unlabeled compound which is called the chase. Now question number 27. A specific protein required for infecting a host by coronavirus is known as. This is the most expected question because for the last 2 years we faced the pandemic of COVID-19. And the option A, receptor protein. B, PDE. C, spike protein. D, polymerase basic 2. I hope you all have got the correct answer, and the correct answer is the option C, the spike protein. The spike glycoproteins, they protrude from the viral surface and attaches with the host cell receptor, and hence the first step of the infection process, which is attachment, is completed. The question number 28. The most common initial step to separate water soluble proteins from insoluble cellular material is A, electroporation. B, differential centrifugation. C, density gradient centrifugation. D, Western blotting. And the correct answer is the option B, which is the differential centrifugation. The particles of different cell density, different size and shapes, they are separated by the method differential centrifugation. The next question number 29 is, the pH at which net charge of protein becomes 0 under an electrical field at room temperature is known as A, isoelectric potential. B, isothermal point C, isomeric point D, isoelectric point. And the correct one is the option D, which is the isoelectric point. It is that page of the solution at which the net charge on a molecule is 0. Here such molecules are considered which are called the Zetter ions. They have both the positive part and the negative part. So here the correct one is the isoelectric point. Question number 30. The microscope that converts slight differences in the refractive index and cell density into easily observed difference in the light intensity is known as A. Phase contrast microscope, B. Bright field microscope, C. Dark field microscope, D. Transmission electron microscope. The correct one is the option A, which is the phase contrast microscope. In the case of the phase contrast microscope, what happens? There are small phase changes in the light rays, and these phase changes, they are induced because of differences in the thickness and the refractive index of the different parts of the object. These phase differences are then transformed to the differences in the brightness or the light intensity. Now, question number 31. Which of the following histone protein found in chromatin has high lysine? The options are H1, H2A, H3, H4. So now what are histones? I know that you know all about histones. Histones, they are highly basic proteins which are found in the chromatin fibers. Their amino acid composition, it varies, and the histone protein which has high lysine is the H1. So the option A is correct. Now the question number 32. Which of the following techniques may be used for identifying a regulatory protein that acts by binding to specific sequence of DNA? A. Southern blotting, B. Northern blotting, C. Southwestern blotting, and D is the western blotting. Here the option C, southwestern blotting, is correct. Now southern blotting is the labeled DNA technique in which the labeled DNA which contains the putative regulatory proteins, they are incubated with the protein extract. It is combined with the western blotting, the resulting DNA protein complexes, then they are separated by the gel electrophoresis. The option C, the southwestern blotting, is correct. The question number 33. Which of the following disease in humans is caused due to expanded microsatellite repeat? The option A, Huntington disease, B, cystic fibrosis, C, tuberculosis, and D is alkaptouria. Here the option A, the Huntington disease, is correct. In the case of this disease, there is a gene, the gene which is the HD gene. This gene contains the repeats. The repeats, they are the small repeats. These repeats are the repeats which having the sequence CAG. The 5' to 3' sequence is the CAG. And these repeats, they are repeated for 6 or more than 6, it can be up to 35 times. And because of this repeated CAG, a lot of protein is produced. That protein is the glutamine protein. So because of which there is this disease. So now the question number 34. Which of the following disease is associated with Philadelphia chromosome? The options are A, retinoblastoma, B, atrial septal defect, C, chronic myelogenous leukemia, D, myotonic muscular dystrophy. Now here the option C, which is chronic myelogenous leukemia, is correct. Now what helps us in the case of the Philadelphia chromosome? Philadelphia chromosome is present in the patients which have the myelogenous leukemia, and it contains an abnormal chromosome which is due to the translocation. So here there is the translocation of the gene. That gene is the ABL gene. This gene, it moves from one chromosome to the other chromosome. It moves from the 9th chromosome to the 22nd chromosome, and this leads to the case of the myelogenemia leukemia, where there is the abnormal proliferation of the B lymphocytes. Question number 35. A classification system that groups organisms together based on the similarity of their observable characteristics is known as. This is a very simple question. I know all of you will answer it correctly. The A is phylogenetic system, B phenetic system, C phyletic system, and D is the complement system. So here the correct one is the phenetic system. The phenetic is based upon the phenotypic characteristics, the observable characteristics, while phylogenetic is based upon the evolutionary characteristics. The question number 36. A class of immunoglobulin present in the serum which neutralizes toxins, activate complements, and crosses the placenta to protect the fetus and neonate is A. IgT, B. IgE, C. IgG, D. IgM. So the correct option is the option C, which is the IgG. The question number 37, another simple question is, the blood group antigens differ in their A. glycosylation pattern, B. phosphorylation pattern, C. methylation pattern, D. ubiquitination pattern. So here we know that there are the blood group antigens. These are the AB antigens, and there is also the H antigens. The H antigen present on all the surface of all the RBCs, while some RBCs they have either the A antigen or they have either the B antigen, or some RBCs they have both AB, while other RBCs they lack both A and B. These antigens they differ in their glycosylation pattern. So the option A is correct. These antigens, these are the oligosaccharides, and here the glycoproteins and the glycolipids they are differing. So their glycosylation pattern is different. Now question number 38. The replication on template strand always proceeds in 5' to 3' direction. If the sequence of template strand is 5' A, T, C, 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 T, 3', how would the correct sequence of newly synthesized strand of DNA be read? So here number of the sequences they are written as A, C, G, G, A, T, and the other sequences are written. So now what you will do, you know the complementarity of the nitrogenous bases. So when A is replicated, there is T. When T replicates, corresponding to it, there is the A. So here the sequence which is corresponding to A, T, C, C, T is the sequence which is the C sequence. So the correct option is the C, T, A, G, 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 A. So the option C is correct. The question number 39. Which of the following conditions is not essential to maintain Hardy-Weinberg equilibrium in a population? A. Large randomly mating population. B. Small randomly mating population. C. Absence of gene flow. D. Absence of natural selection. Now what does the Hardy-Weinberg principle states? It states that when there are two types of alleles, we know for every gene there are two alleles, then the both allele frequency and genotypic frequency they are constant from generation to generation in a large random mating population. So here the option B is incorrect because it is saying small randomly mating population. Now question number 40. Bacteria produce an enzyme methylase which mainly plays a role in A. Modification of DNA, B. Cutting of DNA, C. Repair of DNA, D. Rearrangement of DNA. The option A is methylation of modification of the DNA is correct. The enzyme methylase, it adds methyl groups to the DNA molecule and specifically to cytosine or the adenine. Now the question number 41. In anaerobic conditions, yeast consumes more glucose than in the presence of aerobic environment. This phenomenon is known as A. Hatch-Slack effect, B. Emerson enhancement effect, C. Warburg effect, D. Pasteur effect. Now this is actually the Pasteur effect, so option D is correct. Now what does this Pasteur effect says? This Pasteur effect, it says that there is an inhibiting effect of the oxygen concentration on the fermentation process. Sudden change from anaerobic to aerobic process results in the lesser fermentation. So in the aerobic conditions, the glucose consumption is high, the ethanol production is less. So here the option D, the Pasteur effect, is correct. Now question number 42. Which DNA repair system is affected in the Lynch syndrome? A. Nucleotide excision pair, B. Base excision pair, C is the mismatch pair, and D. Repair of double strand breaks. The option C, the mismatch repair, is correct. The individuals which have the Lynch syndrome, they have a mutation in the DNA mismatch repair system. This repair system normally corrects the errors which can occur during the DNA replication. Now the individuals which have this Lynch syndrome, they have a high risk of developing the cancers, the colorectal cancers, the endometrium cancer, the ovarian, stomach and the pancreatic cancers, and the cancer of the urinary system. So here there is some errors in the case of the mismatch repair system. Now question number 43. Which of the following animal has a pseudocoel? A. Ascaris, B. Fasciola, C. Biroi, D. C. Anemone. Now what is a pseudocoel? Pseudocoel is a cavity which develops between the endoderm and the mesoderm in the adults. Such animals, they are called the pseudocoelomates, and the example are the roundworms, Nemathe. So here the option A, Ascaris, is correct because it is a member of the Nemathe. Now next question, the question number 42, 44 again it is from zoology part. So the following is not a characteristic feature of bony fishes? A. Presence of paired fins, B. Laterally compressed body, C. Presence of homocircle tail, D. Presence of placoid scales. The bony fishes, they have the ctenoid scales. These bony fishes, they are also called osteichthysis. They have the bony skeleton, they have the cycloid and the ctenoid scales. So here the option D is incorrect. They don't have the placoid scale, so it is incorrect. Question number 45 is the method which was first developed for gene mapping in bacteria was A. Transformation in the year 1928, the conjugation in the year 1946, transduction in 1952, and transformation in 1944. The correct one is the B option, it is the conjugation in 1946. So these conjugations were performed by the interrupted mating experiments carried out by Wallman and the Jacob. So the option B is correct here. Okay, now next the question is the question number 46. The question number 46, just wait a minute. The question number 46 is the gold standard method used for detection of the SARS-CoV virus is. Now it is the A. ELISA, B. RT-PCR, C. microarray, and D is the fish. The standard method is the RT-PCR. So the option B is correct here. It is the gold method based upon the reverse transcriptase polymerase chain reaction. The question number 47, which of the following phytochemicals produced in plants defend against herbivores? A. Terpenes, B. Lignin, C. Flavonoids, and D is tannins. The correct one here is the D, tannins. What are the tannins? Now tannins, these are the polyphenols. Tannins are less digestible and they are also bitter in taste. The question number 48. The rate Next is the question number 64. Which of the following antibody can cross the placental barrier? This question is repeated here. It was also previously asked. So here the options are A. IgG, B. IgM, C. IgE, and D is the IgA. The answer which is correct here is the option A, which is the IgG. The IgG isotypic antibodies except the IgG2. These are the maternal antibodies which can cross the placenta, so they can give the protection to the fetus and the neonate. So these were the important questions. In the next part, the rest 25 questions will be answered. Stay tuned. Best wishes for your exams.